Hello and welcome to Phuket Pulse GED Express Screencast with me, Teacher Marky. Today's lesson focuses on meiosis. This is still under GED Science, specifically in biology. On the previous screencast, I talked about mitosis, which is primarily essential for repair of damaged cell. Now, at the end of this discussion, every one of you is expected to enumerate, describe, and explain the different stages of mitosis, and you should compare meiosis with that of mitosis. Now, we start with the definition of meiosis. Now, this is preceded by interphase, which includes chromosome replication. And this is also known as reduction division. And on this case, the original cell is diploid, or in symbol, that would be 2N. Now, next. During meiosis, the daughter cells contain half the number of chromosomes as the original cell, and it produces from gametes, specifically the egg cell and sperm cell. This is necessary for fertilization to take place. And the kind of meiosis which is taking place on the male testes is known as spermatogenesis, and the kind of meiosis that is taking place on the ovaries of females is known as oogenesis. Now, let's answer the question, why do we need meiosis? Meiosis is needed because it is the fundamental basis of sexual reproduction. There would be no fertilization if there's no meiosis. Now, two haploid gametes are brought together through fertilization to form a diploid. So, one haploid from the male sex organ and one haploid from the female sex organ to make it a diploid. For example, this photo, you can see in there like the sperm cell is penetrating the egg cell, and during that time, meiosis is taking place. Now, we have the first part of meiosis. By the way, guys, for meiosis, it is divided into two divisions. For division 1, we have prophase 1. So it's just like quite similar with mitosis. Like you see there, an early prophase and a late prophase stage. So during the early prophase, as you can see on the photo, the homologous pair are evident and crossing over occurs. Later on, I'm going to explain briefly what crossing over means. And during the late prophase, the chromosome condenses and it's very evident already on the equator of the cell, and then spindle forms, and you can also see the nuclear envelope fragments, which will later on become the spindle fiber on the cell itself. Now, next we have the tetrads formed in prophase 1. This is one of the accomplishment on this specific stage under meiosis 1, prophase 1, which is the tetrad. For example, we have here two pairs of chromosome, and this is known as homologous chromosome. And then at a latter part of this stage, you can see that it joins to form a tetrad, and that tetrad is known as synapsis. Now, I'll explain crossing over. So, crossing over is very important, specifically in meiosis, because there is a unique way of sharing down genetic material and properties, and this causes the type of traits that the offspring will get from the parent cell. Now, homologous chromosome and the tetrad cross over each other. Now, there is a need for two chromosomes to form a tetrad because crossing over will take place on that specific scenario. Now, pieces of chromosome or genes are exchanged and it produces genetic recombination in the offspring. Like this photo. As you can see there, that will give you the unique genetic recombination from the parent down to their offspring. Now, next we have here the next phase. This is still under meiosis 1. We have metaphase 1. As you can see, the homologous pair of chromosomes align along the equator of the cell. And then, after that, we have the anaphase 1. As you can see, the chromosomes separate from each other and goes on separate poles. The homologs separate and move to opposite poles where the sister chromatids remain attached at their centromeres or the centrioles. And then after that, we have telophase 1 under meiosis 1. In this case, you can see there the cleavage furrow, which will separate the two cells to get cells far from each other. And then in this case, nuclear envelope reassembles and spindle disappears because they're about to separate from each other. And then after this, cytokinesis divides cell into two. So that would be the haploid of that cell. So we proceed with meiosis 2. As you can see, we have here gene X, the red one. And on this case, only one homolog of each chromosome is present in the cell. During this time, the sister chromatids carry identical genetic information from which they have carried from the parent cell. So the important thing under meiosis 2 is that it produces gametes with one copy of each chromosome and thus one copy of each gene. Now, let's have the first stage under meiosis 2, which is prophase 2. We have here this one. In this case, since we already have two haploid, the nuclear envelope fragment spindle forms. During this time, you can see that the spindle fiber is about to form and then the nuclear envelope fragments is about to develop. After that, we have metaphase 2. Under metaphase 2, chromosomes align along equator of the cell. So you see there two separate pairs of chromosomes from two cells. And then after that would be anaphase 2, where the sister chromatids are separated from each other and they go to their separate poles or to the centrioles. Now the sister chromatids separate and move to opposite poles during this specific stage, and there's none that is being left out on the center or on the equator of the cell. We have telophase 2. For telophase 2, the cells are being separated from each other. As you can see, the cleavage furrow is already evident and they're about to be separated from each other. And during this time, the nuclear envelope assembles and the chromosome decondenses to become chromatin, and then spindle disappears, and then it follows by cytokinesis, where it divides cells into two, like this one. Now, result of meiosis would be this one. There would be gametes like egg and sperm cell, and there would be 4 haploid cells with one copy of each chromosome and one allele of each gene. Now, different combination of alleles for different genes along the chromosomes, as you can see on the photo to your left. Now, let's have a quick comparison on mitosis and meiosis. For the number of divisions in mitosis, there's only one. And for meiosis, there is 2 divisions, like meiosis 1 and meiosis 2. For the number of daughter cells in mitosis, there would be 2. And in meiosis, there are 4 because it has 2 divisions. Now, are the cells being produced genetically identical? In mitosis, yes. But in meiosis, no. Now, for the number of chromosomes, in mitosis, they are the same as compared with that of the parent. However, in meiosis, there are half of that which they get from their parent. Now, where does mitosis taking place? That would be for somatic cells, and in meiosis, that would be for germ cells. Now, when does mitosis takes place? That would be throughout life, and meiosis takes place at sexual maturity. For the role, mitosis is for growth and repair, and meiosis is essential for sexual reproduction. So you can see here a great comparison between mitosis and meiosis. I hope you take note of that. And since you already have an understanding of what I have discussed today, let's try these examples. Now, can you try labeling each stage from meiosis and identify what division of meiosis is this? Is this meiosis 1 or is this meiosis 2? So the first photo is an early prophase 1 where the chromosome number doubled, and then after that we have the late prophase 1. So we have the pre-stage and the post-stage of prophase, and then the third photo would be the metaphase 1. You're right. How about number 4? That would be anaphase 1. And then the last stage would be the telophase, wherein diploid cells was produced, and telophase 1 is being followed by cytokinesis. And what division of meiosis is this? You're right, it's meiosis 1. How about this photo? Can you try to identify which stage of meiosis is that? So on this case, this would be the second part of meiosis. This would be reducing chromosome number. So from prophase 2, that would be followed by metaphase 2, and then followed by anaphase 2, and then telophase 2. The result would be 4 identical haploid cells. Great job! Now, congratulations for getting this so far in my screencast. Good job, everyone. And for the references that I have used for this screencast, we still have here Reading Essentials for Biology, Glencoe 2017, and Glencoe Biology McGraw-Hill Education. If you want to read in advance, you can make use of these references. I'm sure it will help you a lot. Now, thank you so much for watching, and I hope this video is helpful. Since you are already here, you might as well subscribe to our YouTube channel or click the like button and share this video if you want to help your other friends understand the topic that I have just discussed. You can also connect with us at www.phuket.org or go to our Facebook page at facebook.com/PhuketPulse. Or if you have questions or inquiries regarding our organization, you can just give us a message at line or call us directly at 081-417-0978. Thank you so much, and I'll see you on my next screencast. Goodbye, everyone!